This is a video discussing the use of the online resource, the Protein Data Bank, or the PDB. This is a very, very powerful resource, and I'm going to be discussing just a few things you can do with it. But again, as I've stated before, play around with these and look at all of the resources that you can have access to. We're going to be using the search function here. Now you can search proteins by their name, or if you know their protein database code, you can put it here. For your assignment, I've given you a code number that you can just look up, and I'm going to also be putting in a code number. This should bring you to the protein of your interest. In this case, I'm looking at the protein lactate dehydrogenase, and it has some molecules bound to it. You can get information about the protein here, and this particular actual web page is the one that you should be utilizing for your citation. This is the DOI of that citation. You can see the method that was used to actually find out the structure of this protein, the authors that inserted this information, and the organism in which it is normally expressed. This is a protein that is found in humans. If there are any papers that were published that cite this information of the protein structure, they'll be listed down below. You can also find out other information about the protein here. It will tell you things like the molecular weight of the protein. Notice this is in something called kilodalton. This is a little bit different than what you might have seen in the past with just that MW molecular weight. But it is a similar number. We'll talk more in the future about Daltons and what that means. You can see how many atoms are in this protein, a lot. And this tells you actually how many amino acids are in the protein. Now, most of this assignment is going to be dealing with looking at the structure of the protein. So here in the 3D view, if you click on structure, it will bring you to another window that gives you much more control and ability to understand this protein at a more unique level. Up here at the top is the sequence of amino acids that make up this protein. You can see I'm scrolling over them, and in the bottom corner, it tells me information like the actual number in the chain. So this is a leucine. We don't have to worry about the name so much now, but this is a leucine, that's the LEU, and it is number 188. I can also tell that it's number 188 because of these numbers at the top. So this is amino acid number 5, and you can see there's a 5 over it. This can kind of help you determine about where something might be. If I'm looking for amino acid number 200 per se, I can see here's 195 and here's 205. So 200 has to be somewhere here. I can kind of scroll around until I find 200. So that's how you would do that. So aside from the information about a string of amino acids in a chain, the main thing you can do is look at the three-dimensional structure of this protein. You can click and drag to move it around and kind of get different angles. That's what I'm doing right now. And you can also do things like change how you view the protein we're going to be doing some of that in this window right here. Right now, this protein is in a type of structure called a cartoon structure. You can see that word right here, cartoon. That's what's giving it this kind of coil and spiral look. Proteins don't actually look like this, but this is just a way that we as scientists will visualize proteins to kind of understand what's going on with these amino acids. I want to change this to get a more realistic view of what this protein would look like if I was just looking at it. To do that, I'm going to click on my polymer that says cartoon and click these dots here. And this is going to open up a bunch of information. This lets me edit a bunch of things. But mostly here, you'll see something that says cartoon representation. I'm going to click on that. And I want to change this type from cartoon to something different. So if I click on cartoon, it will let me change this to a bunch of different types of views. And you can look at some of these. For example, ball and stick. I'm going to update it. That's what ball and stick looks like. Gaussian surface. This is the main one that I'll be asking you to look at. This gives you a much more realistic view of the protein. It gives you that globular view. It does not look as pretty or fun, but this is more realistic in the way that the protein would look. And there are a bunch of other different types of representations you can pick. Now, one last thing I want to do here is change the color scheme. You can see right now that in my same box, I have information about the color theme, and I'm going to change that. So down here is my color list. And if you open that up, you'll see a bunch of choices, and this is for you to decide what you want. But you can pick one of these other choices of colors, and it will change your protein to be that particular color set. This is how you change the set colors that it's utilizing. Now, what if I'm interested in looking at something more in depth? As in one particular amino acid along the chain of this protein. Well, as I said, if you go along and you find an amino acid of interest, so in this case, I'm going to pick this leucine and zoom in. So I just clicked on it. I clicked in my list at the top, and it zooms in to where it is. And actually you can see I'm not clicking anymore, but I'm just kind of highlighting, and it's showing me where those amino acids are along the chain. So here's my leucine. I'm actually gonna move the structure a little bit so that it can show me that leucine a little bit better. There it is, highlighted in red. And you can see it's actually participating in some non-covalent bonds with other amino acids. And if I want to know what other amino acid is, I can just scroll over again, not clicking, scroll over the other regions. And in the bottom right-hand corner, it will tell me this is a PHE 76. Let's say phenylalanine number 76. And you can see there's something going on with this phenylalanine and this leucine. So that's how you can find out more in-depth information within the structure of the protein. To zoom out, you can use your mouse wheel, or you can, if you're using a touch screen, just use the pinch to zoom out. And you can also just click an empty space that will also zoom you out to see your main protein. Okay, well, that's it as far as information regarding how to use some of the simple functions of the PDB. There's a lot you can do with this editing function on the side. And again, I implore that you play around with it and just kind of have fun. You can change a lot of things. You can toggle things on and off and look at different things. But these are the main functions you'll be using for this particular worksheet. When you're ready to take images and you want to save something that you have on here, you can click this camera shutter button in the corner, and it will let you download a picture of that image, whatever you're viewing at that time. So even if you zoom in on something, you can take a picture, and there you go, you can just download that image.